In this episode, we will talk about how to mimic human metabolism in mice.
For example, neurons, glia.
Let's talk pharmacokinetics for antibody development.
Following the transplantation of hematopoietic cells.
Let's talk how to test if your drug induces a cytokine storm.
Welcome to JAX Tech Talk with the technical information scientists of the Jackson Laboratory.
Hello and welcome to the JAX Tech Talk. My name is Neeraj Tiwari and I work as a technical information scientist at the Jackson Laboratory. My job as a technical information scientist is to serve research community with education and practical solutions. JAX Tech Talk is a 15-minute chat that may be of interest to anyone in preclinical research and drug development. In the past, we have discussed how to create new mouse models using knockout, knock-in, and genetic humanization strategies. Today, we are going to discuss how to create new models using large insertions, some challenges around it, and a new technique that can be employed to overcome the problems with large insertions. So you can post your questions related to this topic during the talk, and we will try to answer them at the end. So to discuss today's topic with me today, I have my colleagues, Dr. Don Tanamachi and Dr. Lizzie Axton. Don and Lizzie, could you kindly give our audience a brief introduction?
Hi, everyone. I'm Don Tanamachi, your Model Generation Services Program Manager. I've been with JAX for about 3 years, but as many of you joining, I've been using various JAX mice and services for a number of years during my academic years and then at various commercial entities where I led model generation design and transgenic core facility teams. I'm currently overseeing all aspects of the model generation program at JAX, ranging from developing and defining what genetic model generation services we provide to and supporting your JAX representatives, which I will pitch off to Lizzie now.
Thanks, Don, and hi, everyone. Thanks for joining us today. My name is Lizzie, and I am a scientist who supports our sales team around our model generation services. I've been at JAX for about 4 years now. I actually started in TIS working with Neeraj, and I consider myself just a general expert on mouse model generation and just working with mice in general. My personal mission in my current role is really just to make mouse model generation more accessible to everyone. So even if you're not an expert, you can still, get the mice that you need for your research. Yeah, looking forward to our chat today.
Thank you so much, Don and Lizzie. So let's begin with today's discussion. Don, can you explain this to our audience? What are large transient insertions and why these are required for creating new mouse models?
Sure. So over the years, using mouse models have become more applicable and powerful as we're learning how to better manipulate the genome. Especially now with gene therapy and gene editing therapeutics, there's a need for larger modifications in a controlled, directed manner. Specifically, there's a growing need to generate more complex and larger humanized alleles, either by inserting or swapping larger genomic regions, or insertion of larger reporters or tissue-specific drivers. So historically, we're initially only able to do simple, random, often chemical mutagenesis, or knockouts, or inefficient random insertion of large transgenes.
So Don, you mentioned large transgenes. That brings us to sort of our first topic of what exactly is large? What does that mean to people? And what size can we accomplish with those random transgenic insertions?
So with the random insertions, we can introduce quite large DNA constructs, such as large plasmids. I've worked on 80 to 100 kb transgenic models, BACs, which usually range from 1 to 200 kb, and even JAX, where I've actually tried to put in several hundred kb to almost a megabase. So the problem with these are the challenges that they go in randomly anywhere in the genome. And you can get, they can go into multiple copies in tandem repeats, with some of the copies going in like fully intact, but you can also have deletions and other abnormalities or aberrations. Depending on where the constructs go, you can have overexpression, altered expression, silence in the transgene, or even in the endogenous region where the transgene inserts, you can actually have deletions. So one thing that's really important is that with these random transgenes, every unique insertion site or every founder line needs to be characterized before we can reliably use that as a representative mouse model. So even though you have the same construct, each founder line is going to be different. So as the demand for more complex genetic modifications arose, we really needed to develop some more elegant technology to modify the genome and manipulate larger DNA sequences.
So Don, based on what you have just now explained, random transgenics has own advantages and drawbacks. And that's why there was a need to develop more targeted approaches to make these insertions, such as zinc finger nucleases, ZFMs, and TALENs, and later on development of Crispr. So Lizzie, can you tell us more about CRISPR and how CRISPR has changed targeted gene editing?
Yeah, definitely. So CRISPR was a really exciting development when it first came out because it was a very simple and customizable approach to, say, more precisely edit genes. So, it's not randomly inserting a transgene into the genome. You're saying exactly where you want to make an insertion. Now, specific to mouse research, one really nice benefit of it is that you can do CRISPR modifications on a bunch of different types of genetic backgrounds. Back in the old days, we would make all of our modifications on, ESLs, particularly our 129 genetic background, and then you had to backcross for like 5 to 10 generations to get it on the background that you actually want. But now with CRISPR, you can make, I would say, small to medium insertions on, the genetic background that you actually need for your research. One particular type of modifier that I think is really powerful with CRISPR is in genetic humanization. So for example, you can insert, a human gene, the cDNA of that gene, directly into that specific mouse locus, creating sort of a simultaneous knock-in, knock-out to genetically humanize that gene. And with CRISPR, you can do it in, have mice to work with in about 8 to 9 months, whereas with other techniques that would take you a couple of years to get to that point. So yeah, very highly adaptable, precise and efficient method. The real limitation, especially in this conversation, is just the size limit. So, at JAX, we still do a lot of our modifications with CRISPR, but we usually only use it for up to insertions of about 5 to 6 kb, because beyond that, the efficiency just starts to substantially decrease. So we need different methods for basically more targeted large insertions.
So if I can add just one little quick thing too, because with CRISPR, you're still actually using homologous recombination. So that's actually one of those things where you actually can have other modifications that you don't want.
So in case we don't want these large size insertions to be random, and as Lizzie explained, there is the size limit of how much we can actually insert using CRISPR. What do we do if we want better control on the binding and expression of these large size fragments? So Don, can you tell us other tools which are available that can help us with large insertions to be more targeted?
Sure, I would love to. So there's a recent JAX publication that describes the use of the bacteriophage BXB integrase, which can accomplish large single copy insertions. So as I kind of butted into Lizzie, but BX works differently than homologous recombination. In a sense, it's actually using an integrase, and it's utilizing at or attachment sites. So these flank the DNA that's going to be inserted, and they match with the at landing pad sites in the genome. So what we have is the researchers at JAX have developed, designed a unique dual directional landing pad system, so larger insertions can integrate directionally as a single copy. So there is no CRISPR Cas9 mediated cutting of the target genome. There's the reduction or basically elimination of the Cas9 mediated off-target insertions. You don't actually have to deal with that homologous recombination over large regions, where you can actually get recombination into different areas or intra-chromosomals, interchromosomal regions. That's what actually happens a lot with the larger pieces.
So it's a targeted insertion. So can you put a BXB insertion anywhere in the genome, or is it specific locations?
So you can. What we actually have at JAX is these dual at landing pads have been positioned in the Rosa 26 locus. So the Rosa 26 is one of those safe harbor open loci, And what this means is that the region is transcriptionally open and active, permissive for expression under the control of whatever promoter or transcriptional cis element or whatever you want to put in there. It's like a free space in the genome. So you're actually going to only what you put in is what's going to get transcribed or under the direction of.
Nice thing about Rosa too is it just has strong widespread expression in the mouse. So if you want something that expresses basically in the whole tissue or combine it with something like tissue specific promoter, you've got a lot of options there with Rosa.
Yeah, definitely.
So, one thing I was just telling people about is CRISPR works in a bunch of different genetic backgrounds. What about BXB in terms of what genetic backgrounds works with BXB?
So we can actually do BXB, the technology, in a background. However, what we actually have developed right now with this altered Rosa 26 dual landing pads, we actually only have it in a couple of strain backgrounds right now. So it's a B6J. We have a couple wild type flavors, and as I like to call it, B6J black, albino mice. We also have it in, and we're developing NOD and NSG mice. And we're actually further developing different strains and also in zygotes and in the ES cells too, that technology. So, and I'm going to let Neeraj kind of tell us about NSG mice.
Yeah, I'll be happy to explain that to our audiences. So the main reasons for using NSG strain for this BXP1 integrase platform is not all of the audiences are familiar with that. So first of all, NSGs are highly immunodeficient strain. And the advantage of that is it can be used for primary tumor engraftment studies. So it can accept xenografts of some human tumors that do not engraft any other strains. Also, immuno-oncology studies where human immune system can be engrafted and propagated in these mice. So down up to what KB size insertions can be done using BXP1 integrase platform?
It's true. I didn't actually talk about size here. So the paper that we published reports very robust integrations from about up to 10 to 15 kb with several experiments inserting 20 to 30 kb inserts and even a 42.9 kb insert. However, right now, JAX is currently guaranteeing DNA insertion size up to about 10 kb, but we're doing a lot of experiments, so we're actively testing the larger sizes so that we actually can guarantee more bigger sizes, and as I said, working on also different strains as well.
Thank you. Thank you for explaining that to our audience. So Lizzie, can you tell us, our audience, if they are looking to make large insertions or targeted insertions using CRISPR, where they can find this information or whom they can reach out to?
Yeah, definitely. And let me just see if I can.
So while Lizzie pulls that up, I was going to say the timing for the BXP is very similar to the CRISPR. So.
Okay. Can you see my screen just fine?
Mm-hmm.
Yep.
Okay, perfect. So yeah, so at JAX, we do have services around, CRISPR, random transgenics, BXB, you name it now. And one of the best ways to get started is just by reaching out to us. You can contact your JAX representative. You can reach out to me directly, or really anyone at JAX that will get you sorted. I just wanted to highlight a couple of quick resources for you. Over on this side, we have a custom model generation website that has all kinds of information on additional resources for you. A lot of blogs that Don and I have put together over the years and other, live recorded events such as this one. I just want to do a quick highlight of this form of Speak to a Scientific Expert. At least as of right now, that comes straight to me. So if you want to get started, that's a pretty quick and easy way to get your model gen discussion going. Here's just an example of a blog that we did on making genetically humanized mice. And I noticed that a couple of chat questions were coming in.
Yeah.
This is at least a snapshot of the BXP1 publication. And we can also follow up with a link for people who are interested in reviewing that. It did come out, gosh, I want to say about 2 months ago now.
It was the end of March.
Yeah, so it's a couple of places that you can get started. And overall, I just want to highlight, I don't want to leave you all with the impression that you have to be an expert to, make a new model. That's exactly what JAX is here for. So if you come to us with your goals and let us know what you're trying to accomplish, we can sort of point you in the right direction and help you figure out, what strategy might work best for you. So I definitely encourage you all to reach out, and let's see if I can stop sharing.
So let's take some of our audience questions that are coming. So one of the common questions were about the reference, which Lizzie already described in that PowerPoint slide. The next question we are getting from our audience is, how well can we adapt the dual landing pad to any other locus other than the ROSA locus? So Don and Lizzie, anyone can answer that?
Yeah, I can address that. So right now we actually have it in the ROSA 26 locus. We can put it into any other locus or any safe harbor locus. Again, it's just an insertion of those at sites. They aren't very large, so it's not that modification is not very difficult. The other thing too is that we can actually modify, like if you want to do a platform mouse or you want to do kind of an iterative process. So if you have a place where you want to put those at sites in, we can do that, and then you can actually do it iteratively, or you can actually directly compare insertions of different constructs at the same place. So as I said, if you wanted to do it as a random transgene, you have to not only express or compare where to integrate it in the copy, but this way you can actually do it into the ROSA 26 or any other locus. So
one more question we are seeing in our chat box is BXB1 available on all genetic backgrounds?
So as I said, we right now we actually have it on the B6 and the NSG, and there's a few other strains coming out. We can actually modify any other strains backgrounds to actually do that in the ROSA 26. Not that you want to actually do a lot of crossing, but we can actually modify the B6 and then cross in if you have another gene that you want or another modification that you want to cross into it. But we can also directly modify the lines, any strain. Again, it's more of a two-step process in that aspect. So you'd have to actually put it in and then put in your larger transgene. As I said, we're actually expanding our repertoire of how many strains. So just give us some time, and we're developing different strains and bigger sizes.
So what are some of the models that have any clinical significance? Are some kind of like clinical relevant models are also available?
So we do actually have one human ACE2 mouse that was created with the BXC. We actually have several on different backgrounds. So this is just an example where you can actually make that same modification on different backgrounds using technology with the different strains that we actually have. And those are all available. Some of them are pre-ordering right now. Some of them are available for service. So contact Lizzie or our TIS group, and they'll be happy to point you to those mice. So
Lizzie, this question is for you. Does JAX still generate CRISPR or random transgenic mouse models?
Definitely. No, we can use, our current service offerings with JAX are, CRISPR, random transgenics, and then also BXB1 now, we're starting to do those projects as well. So yeah, sort of back to what I was saying around, you don't have to come to us with a specific technology in mind. If you let us know, what you're hoping to accomplish, we can sort of advise on what the best approach overall would be.
So we can take one more question from our Q&A. And one of the questions I'm seeing right now is this BXB landing mouse is commercially available for researchers?
Yeah, I can take that one. So we do have, the original BXB1 mice with those landing pads in the ROSA 26 locus. You can find them using our mouse search tool. I believe at the moment we only have the one on B6J that's actually, a live colony ready to ship, but we also have the ones on the other genetic backgrounds as well.
Yeah, thank you.
I also noticed with one comment, it is clean that there's no vector sequences in the insertion. So that's actually one of the nice things about the BXB.
Yep, good to know. So thank you again, Don and Lizzie. This conversation was really informative and perhaps helpful for you all to have a better understanding of how we can use large insertions to create new mouse models. So that's all the time we have for this week. Thank you for joining us. Our next JAX Tech Talk is episode Let's Talk Reducing Bias in Mouse Studies. We'll be live on Tuesday, July 26th. Follow JAX Tech Talk on LinkedIn so you are notified anytime we have a new episode, or subscribe to our playlist on YouTube and catch all the previous JDT. This is Don, Lizzie, and Neeraj signing off, wishing that you all stay healthy, stay safe, and stay excited about your research. Bye-bye.